Hello everyone, and thank you for joining us. Natasha Sheybani, PhD, will introduce the concept of gene therapy and explain how focused ultrasound technology is poised to play a role in this exciting field. She will review the strengths and weaknesses of gene therapy and provide an overview of preclinical applications in the field to demonstrate the utility of focused ultrasound-mediated gene therapy across a range of diseases and conditions. Dr. Natasha Sheybani is a postdoctoral research fellow at Stanford University, where her appointment is shared among the departments of oncology, radiology, and biomedical data science. Her research focuses on advancing machine learning-based radiogenomics for precision oncology. Dr. Sheybani received her bachelor degree in 2015 and her PhD in 2020 in biomedical engineering from Virginia Commonwealth University and University of Virginia, respectively. Our graduate research at UVA centered on leveraging image-guided focused ultrasound to potentiate immunotherapy for primary and disseminated solid cancers. During her training, Dr. Sheybani was supported by the NSF Graduate Research Fellowship and Robert R. Wagner Fellowship, and she currently holds the prestigious NCI F99K0 pre-doctoral to postdoctoral fellow transition award. Before we get started, a few technical items. If your connection is lost, please simply log in again through the link you received when you registered. You will receive a link to a recording of the webinar as well. If you'd like to ask a question, please submit it at any time via the Q&A function at the bottom of your screen. We will collect these questions and answers as many as possible during the Q&A period at the end of the presentation. And now, I'd like to introduce Dr. Natasha Sheybani. Natasha, the floor is yours.
Thank you so much, Fred, for the introduction. And I would like to thank Neil Cassell and the Focused Ultrasound Foundation for this kind invitation to give a webinar today. In the next hour, I'll be introducing and discussing the exciting interface of two uniquely disruptive therapeutic techniques, gene therapy and focused ultrasound. In this talk, I will first introduce the concept of gene therapy, overviewing how it works, how it has advanced to date, advantages and disadvantages, as well as outstanding challenges. After that, I will introduce focused ultrasound technology and how it's poised to play a role in gene therapy. And finally, I'll close the talk with an overview of a few applications for focused ultrasound-mediated gene therapy. So as I understand it, we have a very diverse audience here today. And so I thought I would start with this talk with a very brief foray into molecular biology by first asking the simple question, what is a gene? So genes are segments of DNA that direct our cells to make proteins, and proteins are molecules that are capable of performing a vast array of functions throughout the body. There are around 20 to 25,000 genes in your genome, and these are made up of approximately 3 billion individual chemical base pairs. And as it turns out, very small changes in DNA can markedly impact how our cells function. In diseases with some sort of genetic basis, it's typically the case that DNA with mutated genes, as shown here in red on the DNA strand, has directed cells to produce abnormal or defective protein, or has disabled production of a protein completely, as shown here. Mutations can be inherited, but they can also arise as a natural byproduct of cellular aging or as a result of environmental factors like exposure to toxic chemicals. And importantly, these mutations can have devastating impacts on normal bodily functions at a macroscopic level. So as such, there are a multitude of diseases underscored by this concept, ranging from rare genetic disorders to major public health concerns like cancer. So with this background in place, let's talk about gene therapy. Gene therapy is the introduction, removal, or change of nucleic acids within a patient's cells in order to correct or prevent a pathological process. Once inside the cell, gene therapies can do things like reduce levels of disease-causing protein, increase production of disease-fighting proteins, or produce new or modified proteins altogether. And in gene therapy, typically what happens is that a functional copy of a gene is delivered into the patient's cell, and then the normal protein can be produced from that functional gene in order to hopefully correct the underlying cause of the disease. So in fact, researchers and clinicians are exploring a multitude of approaches when it comes to gene therapy. So I'm going to quickly overview the many ways in which this can work. The first category here is gene replacement. And here the goal is to replace a non-working mutant gene with a healthy gene. And this can make sense to do for genetically driven pathologies where there's a loss of function, such as Duchenne muscular dystrophy, which is caused by a genetic mutation that prevents the body from producing dystrophin, a protein that muscles need to work properly. Another approach is gene silencing, which is comprised of knocking out or inactivating an inappropriately functioning mutant gene, and an approach where this holds value in quote-unquote gain of function genetic ailments is the example of Huntington's disease. In this disease, a mutation in the gene that codes for the Huntington protein results in progressive degeneration of neurons in the brain. We also have the option for gene addition, which entails introducing and usually overexpressing new genes into cells as a measure of disease protection. And a great example of this, which I'll elaborate on in a few slides, is chimeric antigen receptor, or CAR T-cell therapy, which is a promising form of cell-based therapy that's presently being used to fight different types of cancer. And last, but certainly not least, is gene editing, which enables permanent manipulation of a gene in a patient's genome. Tools like CRISPR-Cas9 fall into this category, and for this reason, this is often referred to as a molecular scalpel or gene scissors. These tools have revolutionized the way that we think about gene editing, as they allow us to add, remove, or change genetic material at specific locations in the genome. And with this comes many important social and ethical considerations that unfortunately fall beyond the scope of my talk today. So before diving in any deeper, I'd like to offer a bit of a history lesson and overview of the field. Nearly half a century ago, prophetic accounts were made by leading scientists about the potential and challenges of using gene therapy to treat inherited monogenic disorders. Interestingly, it wasn't until 1990 that the first clinical investigation of gene therapy commenced at NIH for a rare immunodeficiency disorder. Despite some toxicity-related setbacks that occurred at the field's nascency, gene therapy is now burgeoning, with over 2,500 clinical studies having been initiated since that time. These studies explore a vast array of applications spanning monogenic diseases, infectious diseases, complex neurodegenerative disorders, as well as liquid and solid cancers, and of course, so much more. So the figures here summarize clinical activity in the field of gene therapy up to about 2019. As you can see, the number of approved gene therapy trials has been steadily increasing over the years, with last year's record boasting 232 trials. A majority of these trials are conducted in the US, and moreover, the greatest proportion of these trials is focused on cancer. That said, the pie chart here on the top right illuminates the diverse nature of gene types that are being transferred in these clinical trials, and these include antigens, receptors, cytokines, suicide genes, growth factors, etc. And finally, it's worth noting that over 50% of trials to date have been phase 1, but an appreciable number are at advanced stages of clinical evaluation, as shown in the pie chart here. In fact, between the European Medicines Agency, or EMA, and the US FDA, at least 6 gene therapy products have been approved to date. These include 2 CAR T-cell products for B-cell cancers and 4 other products for serious monogenic disorders, including beta thalassemia, a rare form of vision loss, spinal muscular atrophy, and a rare form of primary immunodeficiency. So how are these gene therapies actually delivered? Well, a majority of gene therapy approaches utilize a specific type of carrier for delivery of a therapeutic gene, which is known as a vector. Appropriate vector design is an imperative consideration in the development of successful gene therapies, which require robust and sustained transgene expression in properly targeted cells, all while avoiding transgene-related adverse events such as off-target or overexpression-associated cytotoxicity or immune responses. The most commonly used vectors are typically derived from viruses, because viruses have evolved in such a way that they can enter cells very efficiently. These engineered viruses, or viral vectors, offer high transfection efficiency and transgene expression that can be controlled, which is beneficial from a safety standpoint. However, this comes at the expense of challenges in vector scale-up during manufacturing, limitations in packaging capacity, immunogenicity, oncogenicity, as well as relative costliness. Meanwhile, non-viral vectors can include naked DNA, organic vectors made up of lipids, peptides, or other natural or synthetic polymers, and finally inorganic vectors, which mainly include calcium phosphates and metal nanoparticles. Non-viral vectors have the benefit of a lower production cost, slightly greater ease of manufacturing, flexibility in chemical modification or functionalization as well. And they also offer a tremendous advantage in biosafety given their lower immunogenicity when fabricated from biocompatible materials. So non-viral vectors also have a higher packaging capacity, but there are still two major constraints that have limited their advancement to date. One of them being their lower efficiency at entering cells relative to viral vectors, and the other being the temporary nature of transgene expression. As shown in the pie chart here, this depicts the proportion of different vectors used in gene therapy clinical trials, and you can probably appreciate that a majority of the work to date has been done with viral vectors. But that said, many advancements toward clinical adoption are now addressing the material safety profile, targetability, and efficiency of non-viral vectors very well, such that they will be an invaluable asset to gene therapy in the coming years. So how are vectors administered? There are really two ways in which vectors can be administered for gene therapy. The viral or non-viral vector containing a genetic payload can be directly administered to a patient either via local or systemic injection. And some of the approved therapies that leverage this route are listed here on the bottom right. And alternatively, cells can actually be collected from the patient or a donor and modified ex vivo. In this case, a delivery vector containing the genetic payload would be used outside of the body to modify the cells, following which those modified cells would be amplified and re-administered to the patient. Examples of approved ex vivo gene therapies are listed here on the bottom left, among which you will notice are CAR T formulations as well. CAR T-cell therapy is actually a great example of an approach that requires ex vivo viral gene transfer, non-viral DNA transfection, or genome editing in order to be generated. And since I'll spend most of the remaining slides discussing in vivo gene therapy applications, I'll go ahead and take a brief moment here to elaborate on the example of CAR T. So CAR T cells, as mentioned before, are a form of immunotherapy that really could revolutionize the way we treat cancer. When properly assembled, CARs can successfully activate T cells and redirect them in an antigen-specific fashion, as shown here in the video that's looping on the left. In a typical workflow, peripheral T cells, as shown here, are sampled from the patient's blood and taken back to the lab, where they're genetically modified to produce chimeric antigen receptors that improve their ability to target and kill tumor cells. After expansion, these CAR Ts are then infused back into the patient. And in an ideal scenario, these CAR T cells will then surveil the body and identify tumor cells expressing the antigen that they're programmed against. Following such an encounter of the right protein, they would latch on and trigger the T cell to destroy the tumor cell. I'll briefly return to the concept of CAR T one final time at the end of this talk. I've previously talked about some of the disadvantages of gene therapy, once again listed here for the audience's reference. And so I'll focus here on some of the key advantages. Gene therapy addresses important unmet needs. It is especially true for rare, debilitating diseases that have few or no treatment options. The approved therapies to date have had high efficacy, and a major advantage is that these therapies aim for a single administration as opposed to repeat or maintenance administrations. And this can vastly reduce or even eliminate the need for other costly treatments while also improving patient quality of life. Another benefit is that gene therapies target the root cause of the disease in many cases. Lastly, it's worth noting that traditional drug design can take years and it can be very costly. Small molecule drugs often have to be screened, developed, and then tested preclinically, following which they can transition into clinical trials, and that process can take anywhere from 10 to 15 years on average. Gene therapies, on the other hand, offer somewhat of a benefit in that they can reduce this window because the drug development phase starts with a specific therapeutic agent, particularly when it comes to monogenic or rare diseases. And this thus allows some bypassing of the drug discovery phase, thereby shortening that time frame I alluded to. Remaining challenges for gene therapy include understanding and preventing genotoxicity from integrating vectors or off-target genome editing, as well as improving gene transfer editing efficiency to levels that are needed for effective treatment of target diseases. Moreover, targeting organs in vivo is very attractive because it avoids a lot of the practical and regulatory hurdles that are associated with ex vivo cell-based gene therapies. However, this still requires that we improve targeting of therapies to the right tissues and subsequently to the right cells. So it's particularly important that we push to make these therapies less invasive through systemic administration while conquering some of those other challenges as well. Finally, with in vivo administration comes the challenge of limiting immune responses that are induced by the vectors. And with all of these challenges comes a tremendous opportunity to involve focused ultrasound in the improvement of gene therapy. So again, I'll open with a simple question. What is focused ultrasound? Unlike the more traditional mode of diagnostic ultrasound that we tend to think of in the context of imaging, focused ultrasound uses a transducer with a curved aperture that enables the propagation and concentration of sound waves into a single ellipsoid volume in a non-invasive and non-ionizing manner. And this energy density at the focus results in a variety of bioeffects within the tissue that can range anywhere from thermal to mechanical in nature. The table here summarizes many mechanisms of action that can result from these bioeffects, and I'd like to draw your attention to the drug delivery column. There are multiple mechanisms that render FUS a well-suited platform for improving drug and gene delivery. And the first mechanism I'd like to talk about is blood-brain barrier opening. So I'm going to show an animation here to just briefly illustrate how this works. What we're looking at is a cross-section of a capillary, which is lined with endothelial cells and tightly sealed off from the neighboring brain parenchyma via tight junctions. As we begin by systemically administering our gene therapy, shown here in orange, we can do that alongside systemic administration of microbubbles, which are gas-filled particles that are typically on the order of 2 to 3 microns in size. These microbubbles can then act as acoustic amplifiers in the presence of an acoustic field that's generated by an extracorporeal transducer. The oscillation, or otherwise known as cavitation, of these bubbles can then result in transient disruption of the tight junctions, as well as other mechanisms of sonoporation and transcytosis. And taken together, it's really these mechanisms that are able to vastly improve the penetrance of a given gene vector into the brain parenchyma. I'll also note that this mechanism is well established clinically. Here I'm showing some images of different FUS devices that either come with or without image guidance. Pictured here we have Carthera's SonoCloud device, which is an implantable therapeutic ultrasound device. And then we have the Neuronavigation Guided Focused Ultrasound, or NaviFUS system. And then down here is the Insightec Exablate Neuro system, which is an MRI-guided FUS system. And as an example of what BBB opening looks like in patients, I'm here showing an image, an MRI image. It's contrast enhanced of a brain tumor patient's brain following focused ultrasound BBB disruption. Hopefully from this inset here, you can appreciate that the region of sonications is apparent on this post-FUS image. There's a clear region of contrast enhancement that's indicative of successful BBB opening. And just to emphasize the translational potential for gene therapy from here, I want to note that there's a growing expanse of FUS-BBB opening clinical trials around the world, and the devices shown here are all being used for multiple disease indications in this context. Other relevant mechanisms to improving gene therapy outcomes include hyperthermia, which consists of subablative tissue heating via low amplitude FUS waveforms. We also have sonoporation, wherein mechanical perturbation with low intensity FUS prompts the opening of pores within cell membranes and allows drug entry into cells. And lastly, we have vasodilation, as it's been shown that FUS can stimulate the release of nitric oxide by endothelial cells, causing muscle relaxation, blood vessel dilation, and a resulting increase in blood flow. And the list that I'm providing here is by no means exhaustive, but simply is meant to capture for the audience the diversity of mechanisms that FUS can potentially contribute to the goal of potentiating gene therapy. So these mechanisms and others that, as I mentioned, time won't permit me to touch on today, have given way to many different applications of FUS for gene therapy. These have included advancements for neurological, ocular, cardiovascular, and hepatic diseases, as well as other diseases like arthritis, as well as various cancers. I'm only going to have time today to scratch the surface of these applications, and I'll thus focus on just a few neurological and oncological applications. Let's begin with gene delivery to the brain. Multiple studies have now demonstrated that FUS-mediated blood-brain barrier opening can improve gene delivery to the brain parenchyma. And here I'm highlighting two example studies that have demonstrated this with reporter genes. The study highlighted on the left is an example of a viral vector-based approach wherein FUS-targeted delivery substantially improved the number of neurons that were transduced by an RAAV vector with a neuron-specific promoter. The transgene expression in the FUS-targeted region is here shown in dark brown, and hopefully you can appreciate that there's much denser staining relative to the contralateral brain that's shown here in the black square. In a different study, we can see that FUS also improves non-viral vector delivery. On the right, we have FUS-BBB opening that was performed in the presence of systemically administered brain-penetrating nanoparticles, or BPNs. And these particles are unique in that they're densely coated with a polyethylene glycol coat that basically promotes the particle's stealth and dispersion within the brain tissue. Per the bioluminescence imaging time course that's shown here, as well as the quantification shown below, delivery of DNA-bearing BPNs across the BBB with FUS yields robust localized transgene expression in naive rat brains, beginning as early as 24 hours after intervention. In a yet different FUS paradigm in the naive brain, it's been shown that endothelial selective or sonoselective transfection of the cerebral vasculature can be achieved independent of BBB opening. This animation shows the overarching mechanism, which is enabled through the delivery of a cationic plasmid-bearing microbubbles with low pressure FUS. This figure shows IF staining of the endothelium and transgene, in this case an M-Cherry reporter. The co-localization of signal here between the capillaries and the M-Cherry transgene suggests that with the sonoselective FUS regime, an increased proportion of endothelial cells were transfected. The figure here shows that this transfection was also confined to the capillaries, as denoted by these yellow circles, with little to no evidence of M-Cherry expression in the arterioles and venules. In the future, this paradigm could have very high utility for endothelial cell-directed gene therapy in settings such as ischemic stroke, where cerebral tissue may already bear a high degree of instability and inflammatory mechanisms such that BBB opening wouldn't be desirable. Now switching gears a bit to some of the pathological setting, I'd like to show a few examples relating to Parkinson's disease, which is a progressive neurodegenerative disorder involving degeneration of dopaminergic neurons in the brain. It's now been shown that outcomes of AAV delivery can be vastly improved with FUS-BBB opening for treatment of Parkinson's disease. And the study that I'm showing here used an AAV-mediated gene transfer of glial-derived neurotrophic factor, or GDNF, in the NPTP mouse model of early-stage Parkinson's. The enhanced portion on T1-weighted imaging, as you're seeing here, T1-weighted MR, I should say, as well as a 3D rendering of that image, show the region of focused ultrasound BBB opening. So for those who are perhaps not seeing what I'm alluding to, the contrast enhanced regions here are where the barrier was opened in these mice. The images here show representative TH staining of the striatum, where you can appreciate the amelioration of dopaminergic projections in the FUS-positive hemisphere, as noted here. And the images on the far right show immunofluorescent TH staining in the substantia nigra. Here we can see much denser dopaminergic projections in the FUS recipient setting as well, and this suggests that the AAVs delivered across the BBB with FUS promoted a stronger effect on dopaminergic dendrites. And what I'm not showing here is that these mice also saw improved neuronal function as exhibited by behavioral testing. So all in all, this study suggests to us that opening the barrier with FUS can potentiate AAV GDNF delivery in early stage PD, curbing the progression of neurodegeneration and improving neuronal function. It's similarly been shown in a 6-OHDA rat model of Parkinson's that the use of non-viral vectors for targeted GDNF transgene expression is improved by focused ultrasound blood-brain barrier disruption in the striatum, as shown here. So this is a T2-weighted image of a rat brain, and after BBB opening, you can see the region of T1-weighted contrast enhancement in the striatum. In this case, the vector is once again a brain-penetrating nanoparticle. The TH-stained images shown here show dopaminergic neuron density is restored in a superlative manner by FUS-targeted GDNF BPN delivery. And that would be the column here on the far right. And this was interestingly conserved across 2 different time points at which the staining was evaluated. But moreover, rats receiving the BPNs in combination with FUS also saw restorations in their locomotor function, as determined by 2 different behavioral tests, one being the apomorphine rotation test, and the other being the forepaw use bias test. And in both cases, the change in motor dysfunction of 6-OHDA rats was consistent with a reduced degree of Parkinsonism following FUS-targeted BPN delivery. I'm now going to shift gears to Huntington's disease for a moment. So in preclinical models of Huntington's disease, it's also been shown that focused ultrasound blood-brain barrier opening for GDNF delivery bears utility. And this particular study utilized liposomes to carry GDNF plasmid DNA, as you can see from the resulting Western blot for GDNF. FUS-mediated GDNF delivery promoted superlative levels of neuronal GDNF expression in the diseased animals after 9 weeks of treatment. By EM48 staining, it was confirmed that FUS-mediated GDNF delivery also reduced Huntington protein aggregates, which are an important indicator in disease progression. Moreover, these changes were corroborated by a significant increase in expression of neuronal survival marker PCREB. Finally, behavioral assessment, as denoted here by the rotorod test, showed some modest improvement in motor function of diseased mice after this FUS-targeted gene therapy. So again, taken together, these results, much like the Parkinson's results, suggest that non-viral and viral gene therapy approaches bear tremendous utility for neurodegenerative disease going forward. So while these were just a few snapshots of preclinical work in neurological diseases, they were by no means comprehensive. And nonetheless, I'm going to go ahead and pivot now over to some oncological applications, starting with one in the brain. Much as we've been able to achieve robust transfection in the naive brain, as well as in the Parkinson's disease setting with non-viral gene therapy, I'm showing some findings that use a similar platform in the setting of glioblastoma. So here, U87 tumor-bearing mice underwent a single session of barrier opening. And here you can again see a substantial increase in contrast enhancement. And subsequently, these mice saw substantially improved transfection following the delivery of a reporter gene bearing brain-penetrating nanoparticle. The epifluorescence images shown here are quantified over on the right. And as you can see, improved transgene expression was also conserved across two different peak negative pressures, 0.45 and 0.55 megapascals. Finally, the representative images on the bottom right here show that compared to systemically administered brain-penetrating nanoparticles alone, focused ultrasound substantially improved the BPN penetration beyond the confines of the tumor microvessels and into the tissue. Focused ultrasound is also being utilized in peripheral solid tumor models for cytokine transfection as an adjunct to immunotherapy. Here, I'm highlighting a study from Kathy Ferrara's group at Stanford, where they design a paradigm combining low-frequency ultrasound with tumor-targeted microbubbles to partially destroy tumors, as well as transfect remaining viable tumor and stromal cells with plasmid DNA encoding interferon beta in order to trigger an immune response that cooperates with immunotherapy. And this approach leverages the mechanism of sonoporation that I briefly introduced in a prior slide. The bioluminescence images on the left show that this regimen yields localized transgene expression that is tunable with ultrasound pressure. So at a lower pressure of 200 kilopascals, you get appreciable transgene expression, and then this is qualitatively heightened at a higher pressure of 500 kilopascals. More importantly, when combined with PD-1 blockade therapy, this IFN beta directed gene therapy with targeted microbubbles yielded both local and systemic cytotoxic T cell responses, as denoted by an enrichment of CD8 positive T cells in both treated and distant tumors. And this was also corroborated by tumor growth restriction in the treated and distant tumor settings as well. Gene therapy approaches such as this are of increasing importance to the field of FUS immuno-oncology, wherein most efforts are directed at inciting T cell responses with FUS in order to potentiate immunotherapy approaches. So briefly, I'd also like to highlight that the mechanical FUS regimes I've talked about today are not the only options that hold promise for improving gene therapy. Here I've highlighted a few studies that have evidenced a role for FUS-mediated hyperthermia in the spatio-temporal control of gene expression, which could hold profound implications for gene therapy approaches down the road as well. In a similar vein, there are some exciting next generation tools coming down the preclinical pipeline that leverage the idea of using FUS as a tool for gene delivery in optogenetics, as shown here, acoustogenetic control of CAR T cells via ultrasound controllable genetic circuits, acoustic transfection methods, and finally sonogenetic control of ultrasound sensitive ion channels for interrogating intact brain circuits. While I can't elaborate on all of these approaches further today, I just wanted to close by saying that these tools are truly pushing the boundaries of how we can leverage FUS in the context of remote, non-invasive gene control, and these thereby serve as more evidence for the promising intersection of ultrasound and gene therapy. So to conclude, gene therapy has certainly come of age, as it's being said, and promises to revolutionize the treatment of many diseases. FUS can also non-invasively overcome key challenges that are constraining gene therapy at the moment, and it's thus poised to play an outstanding role in gene therapy as well. Preclinical activity at the intersection of FUS and gene therapy, as I briefly touched on today, is burgeoning. And so this is a really exciting time to be evaluating opportunities at the intersection of FUS and gene therapy. And with that in mind, this diverse array of preclinical developments and opportunities is really generating a robust pipeline for future clinical translation opportunities to come. So with that, I would like to thank all of you for your attention, and I'm happy to take any questions.
Thank you, Natasha, for this very, very interesting presentation. And now we will proceed with the Q&A part of our webinar. So the first question we have is about delivery and transfection. Does FUS only improve the delivery to the target site, or can it also enhance transfection?
Do you mind repeating that one more time, Fred?
Sure.
Sorry.
Does focused ultrasound only improve delivery to the target site, or can it also enhance transfection?
I think that's a great question. I believe that there is opportunity for both. I think that some of the preclinical studies right now are getting to the point where they can sort of begin to parse that out. And in fact, some of the studies that I showed today, I didn't necessarily do justice to in terms of pointing out that they do indeed improve transfection efficiency with focused ultrasound treatment as well. Now, whether that can be dissected further in terms of the impact of drug delivery increases versus a pure impact of FUS on the vector itself remains to be seen.
Yeah, okay. I have another question about touching about the CAR T cells. So you mentioned the issue of the specificity with the or targeting specificity with gene therapy. Can you elaborate on how you see a role for focused ultrasound in the context of CAR T cell therapy? I mean, besides lymphomas?
Absolutely. I think in the context of solid tumor therapy, there is a lot of opportunity for FUS. So there are certain constraints right now on the performance of CAR Ts in solid tumors because there are difficulties with the trafficking of CAR T cells to those tumors and then the subsequent persistence of the CAR Ts once inside the tumor given immunosuppressive cues within the local tumor environment. So there's still, I think, a drug delivery angle from which we can leverage FUS for CAR T cell delivery. But also with all of the new discoveries in immunomodulation, I think there will be opportunity to understand how certain immunological cues that are potentiated by FUS can then aid in persistence of CAR T cells or even homing of CAR T cells into the tumor microenvironment. Perhaps to your question about the acoustogenetic work that I alluded to, there are also groups right now that are working on being able to remotely control synthetic genetic circuits with ultrasound that's applied exogenously. And so the idea is that you can basically turn on genetic switches that would upregulate the surface expression of CARs and thereby enable CAR T cell therapy to be controlled via FUS in a more direct manner.
Okay, thank you. The next question, what are the implications of FUS plus gene delivery for neurodegenerative diseases in terms of safety, given that the tissue is already damaged? How would that compare to competing technologies like direct injection?
Yeah, so, I think in terms of comparison to direct injection, the most immediate thing I would point out is that if there's opportunity to really answer that question effectively with FUS, then we spare the patient of a rather invasive procedure that direct injection would entail, as FUS can be applied completely non-invasively with the transcranial systems that I showed before. So to the point about safety of gene therapy, there are several clinical studies now that have been published with regard to the safety profile and repeatability of focused ultrasound-mediated barrier disruption in neurodegenerative diseases. So I am certainly not a clinician, but I would say somewhat confidently that we are comfortable opening the barrier in these settings. The question of how that will ultimately interface with gene therapy, I think is one that the preclinical realm will have to address more thoroughly in the coming years. And that's certainly on the horizon. Some people are already working on that.
Okay. Is there a size dependency of what can be delivered, or is there different strategies to think about using focused ultrasound if we want to deliver nanoparticle or viral vectors or different gene therapy vectors?
Absolutely. So I think, given the size of most viral vectors, it is relative to some of the nanoparticle literature that's out there. Blood-brain barrier disruption with FUS is definitely conducive to delivery of viral vectors in terms of their size. I think most AAVs are on the order of like 20 plus nanometers in size. So that's certainly not unreasonable. The nanoparticles that I talked about today are somewhere on the order of about 50 nanometers. And so again, that is a very reasonable size range to envisage as from a translational standpoint, as well as from the efficiency of crossing the blood-brain barrier. In terms of larger sizes, the upper limit I can certainly point out is that our microbubbles, for instance, are on the order of microns, and that is something that will not cross the barrier even after disruption. I have seen other nanoparticle formulations that are like on the order of hundreds, I'll say about 200 nanometers, that I have also seen as being able to cross the barrier. But unfortunately, I'm unable to state an explicit threshold in terms of nanoparticle size.
Okay. The next question, can BBB opening be applied through the whole brain, or is it better for more localized delivery?
I think it depends on the application. Certainly in the preclinical literature, there are papers out there that are doing sort of treatment regimens with a much larger envelope, and this is something that's being built out clinically as well. So the goal is to be able to modulate the volume of BBB opening and have that option when the pathology necessitates a larger volume of opening. I think to the question though, it really comes down to the application. I don't think that clinically anyone has necessarily had their entire blood-brain barrier disrupted yet. I don't know if the clinical constraints would permit that per se, but depending on the application, I think that we could envision room for a variety of BBB opening volumes, depending on the pathology.
And what do we know about the immune response in the brain after first BB opening for gene delivery?
So immune response, I just want to make sure I get this right. So immune response to FUS specifically after gene delivery with FUS.
Yes.
Is that the question? Yeah, so, so one of the studies I showed, for instance, the SANO-selective regime is a great example of one where there was actually not there was a desire to not invoke an inflammatory response. And of course, that was being done with the reporter gene. But what I can say in that context was that with, as an example of transfecting endothelial cells in that case, that paper, which was actually published by my colleague, Katie Gorick in PNAS, showed that there was not an associated upregulation of inflammatory mechanisms following that focused ultrasound treatment. I think broadly speaking, with blood-brain barrier opening, my work and that of many others in the group has shown that within certain contexts, you can get an inflammatory response. This is both in the naive brain as well as tumors. But in the context of specific gene therapies, I don't think that it's necessarily been a regular readout up until this point. And I certainly think that is one we should think closely about in the future.
Well, thank you, Natasha. This was our last question. We'd like to thank you again. This concludes today's webinar. If you would like more information, please visit our website, FUSfoundation.org, or email us at info@fusfoundation.org. Again, this webinar has been recorded and you will receive a link to this recording. Thank you all for joining us today and stay tuned to our newsletter and website for invitations for future webinars. Have a great day.